So none of this was well understood. And this is the first time this has been done in a live mammalian brain ever. So there have been theories as to how this occurred before now. But what we observed, which was surprising, is that not only do astrocytes and microglia compete for phagocytic territories, but even amongst microglia, so there's a dead cell in the middle. There will be 3 different microglia that are interested in the dead cells, but only one of them will actually engulf the cell body. So what's that interaction? So that has never been described before in literature. That there's actually a cell between microglia competition that occurs, where one microglia actually wins, and literally the winner takes all. And also astrocytes also try to compete for phagocytic territory. Now they usually will not remove cell bodies except if microglia is not present. And you know, you could speculate, because microglia, they move around and they can change their shape to mimic that of the dying neuron, that they are kind of more fit for cell body removal for big structures, while astrocytes are kind of a little bit better off at taking up the small debris and little branches, dendritic branches later on. So none of that has actually been described before, like what makes what determines which microglia actually is going to be the winner, and how there is precise boundaries. And so where microglia stops is exactly where astrocytes take off. So they both do have contact. So they both inhibit each other from eating the debris from each other's territory. But at the same time, there's no gap in between. Literally where one stops is where one starts, and they work efficiently to clear everything apart. So it's new. It opens a lot of doors, and it opens questions, more questions than anything else. Like so when you look into neurodegeneration and trauma and aging and early development, like how are these processes changing, and how can we intervene in disease processes?